Hey guys, it's Doc Janelle. Welcome back to my channel. The subject at hand is Human Anatomy and Physiology, and this is the third part of our cell anatomy and physiology video. So hopefully at this time, you're already able to finish the first two videos because a lot of those prior knowledge that we have already learned will be applied to this third and final installment of our cell anatomy and physiology video. We are going to start our topic with whole cell activity. So we've already discussed the anatomy of the cell. We've already discussed the different ways on how transport is done from one cell to the other. Now, what does the cell actually do aside from transport and aside from the different functions of each organelle? As a whole in totality, what does the cell do? So there are three important activities that the cell actually does. The first is gene expression, which is the production of protein from DNA. So at this point, we've already discussed that DNA is stored in the nucleus of the cell. And this DNA not only remains as DNA, but it eventually dictates the production of our proteins. And we've also discussed that what synthesizes these proteins are your ribosomes. And the synthesis happens at the rough endoplasmic reticulum. That is the site of your protein synthesis. Another whole cell activity is cell life cycle. So this will talk about the lifespan of the cell and what happens or the events that occur in the lifespan. And then lastly, we'll be talking about differentiation or maturity. So once the cell has been able to serve its function and its purpose, what happens to the cell afterwards? So that's what we're going to be talking about. Let's start with gene expression. Again, gene expression is the process of producing proteins from DNA. So, as mentioned, DNA molecules are nucleic acids. It's one of the four organic molecules that we discussed when we talked about the chemical basis of life. So, the building blocks of nucleic acids are again nucleotides. And these nucleotides are joined together to form two nucleotide strands. And these two strands will actually interact with each other to form your DNA or deoxyribonucleic acid. More of that in a little while. So just to review, nucleotides are actually composed of different organic molecules. It's composed of a sugar, a phosphate, and a nitrogenous organic base. That's why if you remember, nucleotides or nucleic acids are made up of CHON-P, CHO to represent your sugar, your nitrogen, and then your P for phosphate. Now, DNA and RNA are both nucleic acids. What's the difference between the two? Well, the most simple difference is the sugar that they have. DNA's sugar is deoxyribose, while the sugar of your RNA is ribose. Another difference that they have is its appearance. DNA is a double helix, and RNA is commonly a single strand. So these are the sugars that we have in the nucleotides. The nitrogenous bases, there are four, depending on whether it's a DNA or an RNA. If we have a DNA, then the four nitrogenous bases are thymine, adenine, guanine, and cytosine. But if we're talking about an RNA, we do not have thymine. Instead, we have uracil and then the three others. So here is a good diagram showing us the DNA. So as mentioned, it's composed of a sugar, as you can see here. Sugar on one side, and then as mentioned, two nucleotide strands, sugar on another side. And then we have the phosphate, represented by letter P. And then here, we have the nitrogenous organic bases. It represents your adenine, T represents thymine, G represents guanine, and C represents cytosine. So here, we're able to infer that there is a pairing that happens. A, or adenine, always pairs up with thymine, while guanine always pairs up with cytosine. So those are the pairing. A is to T, while G is to C. Okay? So just so we can be reminded of the proper structures, a nucleotide is the combination of a phosphate, a sugar, and a nitrogenous base. So this is one nucleotide. So if it is paired with another nucleotide on the other side, then you're able to start the foundations of a DNA. So each nucleotide on one strand has a specific bonding pattern to another nucleotide on the opposite strand, as seen in this image and in the previous diagram that I showed you. Again, this shows us that guanine pairs up with cytosine and that adenine pairs up with thymine. So remember that we use thymine since we're talking about DNA. But in the presence of an RNA, we don't use thymine. Instead, it is replaced by uracil. So these strands will then be connected and it will resemble a ladder that will be twisted along its long axis, forming the classic double helix of the two DNA strands. All right? Now, a gene is a sequence of nucleotides that provides a chemical set of instructions for making a specific protein. So basically, when you separate the double helix and you get two single strands, that is already a gene since it is a sequence or a series of nucleotides. So the gene is where we get the instructions for the protein. And later on, you will understand how it happens. So when we talk about DNA and these genetic materials, it is dictated by what we call the central dogma. You will also encounter this when we talk about cytogenetics when you get to second semester of second year. So what the central dogma tells us is that DNA is not directly, protein is not directly produced from DNA. DNA must first produce an RNA, and it will be RNA that will be used to produce the protein. This flow of information from DNA to RNA to protein is commonly unidirectional and is irreversible. This means that we cannot produce DNA from proteins, nor can we produce RNA from proteins. The production of protein using DNA or the central dogma occurs in three stages, starting with DNA replication, transcription, and translation. So let's talk about DNA replication first. Later on, when we talked about the cell cycle, we will again go through DNA replication. So remember that your DNA is housed inside the nucleus. Thus, DNA replication will also happen in the same organelle. So what happens here is that first, your DNA double helix will have to open up. So if it opens up, it will produce two single strands, as you can see here. The single strand is represented by the darker violet strand. Now that we have two single strands, those two single strands will become a template for the new strand, and it will eventually, the double helix will eventually open up all the way to the end and eventually produce two new DNA strands. So that's what happens in DNA replication. Now, let's go to the meat, which is gene expression. So gene expression, again, is the production of protein from DNA. And it involves two important steps from the central dogma. And these are transcription and translation. When we say transcription, from, of course, the meaning of transcribe is to copy. What happens here is that a copy of the DNA is created, and that copy is called mRNA or messenger RNA. Again, in transcription, a copy of the DNA is created, and that copy is called mRNA or messenger RNA. In translation, it involves the use of that mRNA to produce the protein. So let's start discussing them individually. Transcription. Similar to your DNA synthesis, transcription takes place in the nucleus. Why? Because again, we are making a copy of the DNA, and the DNA is housed in the nucleus. So what happens here is almost similar to DNA synthesis, wherein your DNA will open up, not everything, but only a segment of the DNA. Once that it opens up, it will now expose the individual nucleotides, as you can see here, on top and at the bottom. Once that happens, or once the nucleotides are exposed, the nucleotides, or complementary nucleotides rather, will attach to the exposed nucleotides of the DNA. So as we can see here, your thymine, what is the complementary base pair of that? Adenine. So it will connect to that, and it will start a chain of RNA. So your adenine is on your on your DNA strand. It should pair up with thymine. But remember, in RNA there is no thymine, but we have letter U, which represents uracil. So the next nucleotide will be U. And then your cytosine at the DNA will pair up with a guanine, and so on. So this new strand that is being created is what we call mRNA, or messenger RNA. Eventually, this mRNA will exit the nucleus through the nuclear pores. So again, remember, your DNA contains thymine, adenine, cytosine, and guanine. But in mRNA, we do not have thymine, and it is replaced by uracil, and uracil still is a complementary base pair of adenine. So basically, you simply change the T to U. So here is the complete pairing. Thymine pairs with RNA adenine. The DNA thymine rather pairs with the RNA adenine. Adenine pairs up with RNA's uracil, and then cytosine and guanine, and vice versa. So the product of transcription is an mRNA, which will exit the nucleus through the nuclear pore. So again, in transcription, we will make a copy of the DNA, and that copy is the mRNA. That's why it's called transcription. We transcribed a new copy. In translation, this occurs in the cell cytoplasm. Remember, your mRNA exited the nucleus through the nuclear pore. And what produces your protein? Your ribosome. So the mRNA will attach to a ribosome. And where does this happen? This commonly happens in the rough endoplasmic reticulum. Now, your mRNA, which is the copy of the DNA, will be read per 3 nucleotide bases. And when they are read per 3 nucleotide bases, we call that a codon. So let's look at the diagram. On top in blue is the original DNA template strand. And then we created an mRNA in complementary base pairs, no? So thymine will complement with adenine, thymine to adenine, cytosine, guanine, adenine to uracil kasi wala namang thymine sa RNA, instead it's uracil, and so on. Now, in groups of 3, when nucleotides are grouped into 3 in the mRNA, you call that a codon. So, AAG is one codon, UCA is another codon, GUC is another codon. Okay? So, that's how it will be read. That's why it's called translation. This series of nucleotide base pairs will be translated into an amino acid. Okay, so how will that happen? The codon will be paired with an anticodon. Yan, it will be paired with an anticodon. What is an anticodon? These are 3 nucleotide bases which are complementary to your mRNA. And what carries your anticodons are your tRNA or transfer RNA. Again, your codon, which are group of 3 nucleotides, will be read by your ribosome. Once it reads and groups them into codons, the ribosome will look for an anticodon, which will be complementary to your messenger RNA. So if let's say we will find RNA complements for AAG, it will be UUC, right? Because adenine pairs up with uracil, adenine to uracil, guanine to cytosine. So hahanap siya ng tRNA na merong UUC na anticodon. And then together with the tRNA is already an amino acid. Okay? So it will add these amino acids until you produce a polypeptide. So let me show you that image. Yan. So again, your tRNA, it's called transfer RNA because it transports specific amino acids from the cytoplasm towards the ribosome mRNA complex. And this initiates the formation of a polypeptide chain. So again, this is your mRNA, series of nucleotide base pairs, and then this is your tRNA. This is your tRNA. And as you can see, the first 3 letters here are AUG. Follow my pointer, please. AUG. So it will find an anticodon that is complementary to this. So the complement should be uracil adenine cytosine. And here we have it, UAC. And together with this anticodon, this will code for a specific amino acid. And in this case, it's methionine. So it will combine and the amino acid will be added. So all of these other codons will be read and it will be given a specific amino acid until the amino acid chain becomes longer and longer, and that is called a polypeptide. Alrighty? So let me show you this video in order for us to better understand the process.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
All right. So that's it for our gene expression or the production of protein from your DNA. For the second part of our discussion in this video, we will be talking about the cell cycle. Again, when we say cell cycle, these are the events that happen in a cell's lifespan. So when we talk about cell cycle, we cannot do away with talking about reproduction since that is one of the most important functions of the cell. So why do cells need to divide? Why do cells need to produce other cells? Well, number one, in order for reproduction to happen. A baby grows into a kid into a teenager, and a teenager into an adult because of the reproduction of cells. No? Second, it's important for growth and development. And lastly, for tissue renewal, or when we become injured or when we have a wound, of course, in order for the body to repair, it needs to produce new cells. So cell division has a cycle. No, the reproduction or the production of new cells actually goes through a cycle. And the cycle includes two major phases. These are interphase, which is the non-dividing phase, and mitosis, which is the cell dividing phase. We'll be talking about them individually. So, the cell cycle is represented by this image to your right. It has again two phases, which is the interphase and your mitosis. All right? So for most of the cell's life, it will be spent in interphase. As you can see, it's a much longer period or a much longer phase compared to your very small or very short mitotic phase. Your interphase is divided into three subphases. These are G1, and G stands for gap. So in other books, it's called gap 1 phase. Your S phase or synthesis phase, and your G2 or gap 2 phase. Now, what happens in each subphases? In G1, there is normal metabolic activity, normal life, as they say. But what's special about this is that the organelles are already being duplicated in G1 phase. Why? Because interphase is actually a preparation for mitosis. So, again, in mitosis, it is the production of daughter cells. So, even before the cell division happens, everything will already have been prepared. So, in G1, organelles are being duplicated. In S phase, from the word synthesis, the DNA is already being replicated. And remember, we discussed DNA replication earlier. So the S phase centers on the events in the nucleus. The opening of the DNA strand, the production of two single strands, and then the creation of the new complementary strand in order to have two DNA sets. And then G2 is the cell preparation to divide. So at the end of interphase, a cell already has two complete sets of genetic material and two complete sets of organelles. Okay? Now, again, we have your G1, or where there is normal or routine metabolism, will be followed by your S phase or DNA replication, which happens in the nucleus. And then G2, which is a preparation for division. It's a much longer phase compared to your mitosis. Now, before we go into mitosis, let's review a little bit of information. Human cells, and this refers to every single cell in the body except for sperm and egg cells. So all human cells contain 23 pairs of chromosomes. for a total of 46 because 23 pairs. Now, out of the 46, 23 comes from our mom and 23 comes from our dad. Okay? That's why when the sperm and the egg cell unite, the sperm from our dad and the egg from the mom, when they unite, it gives a total of 46 chromosomes. Thus, it tells us that sperm and egg only contains half, right? It only contains 23 chromosomes. So again, sperm and egg only contains 23 and we call that haploid. And when the sperm and egg unite to form a human organism, and the human organism is made up of several cells, all these cells already contain a complete set of 46, which we call diploid. So, one of the 46 chromosomes, one pair rather, of the 46 chromosomes are sex chromosomes. Okay? So, 44 will be human chromosomes, and then 2 will be sex chromosomes. If the chromosomes are X and Y, then the result will be a male organism. But if the chromosomes are 2 X's, then the organism will be a female. Now, mitosis involves the formation of two daughter cells from a single parent cell. So here we have a mother cell, and at the end of mitosis, it will be able to produce two daughter cells, two identical daughter cells. I say identical because the DNA that they both have came from the same DNA. And mitosis usually is divided into four phases: prophase, metaphase, anaphase, and telophase. What we're going to do now is to discuss each and every phase of mitosis, starting with prophase. So during prophase, the chromatin, which is, we discussed this when we talked about the nucleus in the second video. Chromatin is the loosely arranged formation of DNA and protein, if you still remember. During prophase, that loose arrangement will become condensed. And from chromatin, it will become this structure here called chromosome. So this is how a chromatin would actually look like. It's very loose. The nucleus is completely filled with all of this DNA strand. But then in prophase, from a very distributed appearance, it will become like this, very condensed. Alrighty? It will become your chromosomes. Aside from that, during prophase, your centrioles, these are your centrioles, they will start to produce what we call spindle fibers. And the spindle fibers are actually made up of microtubules, no? So what the centrioles will do, they will go to opposite sides of the cell. Okay? One will go to this part, another will go to this part. As you can see in the original formation, they are next to each other. But then in the next picture, they started to go to the opposite sides, no? And then we see here, there's spindle fibers. Also, another thing that we'll notice is that the nuclear membrane starts to dissolve. This is the original image, and here we see that the nuclear membrane is starting to dissolve already. Now, why do I have this image? Because I want to show you that the spindle fibers actually attach themselves to the kinetochore, which is found at the centromere. Whoa, I know. Wait, let me explain that to you. The kinetochore is attached to each single chromatid. Okay? And then the centromere is the middle region where these two are connected. So, ang ibig kong sabihin, yung kinetochore, naka-attach siya sa isang chromatid. Ito kasi chromatid to mag-isa. And then, ito ay twin niya, no? Kaya nga sila sister chromatids. They are identical. At sila ay pinagdikit sa centromere. Okay? So, yung spindle fibers natin, mag-a-attach sila sa kinetochore natin. Okay? Kinetochore. So, as a summary, again in prophase, there is condensation of DNA to form chromosomes. Number 2, we see that the centrioles start to go to opposite ends. And number 3, we see that the nuclear membrane starts to dissolve. Those are the 3 important events in prophase. Second, we have metaphase. In metaphase, there is only one event that happens here, which is the alignment of the chromosomes at the center of the cell. And we call the center the equatorial line, similar to how the equator is the center of the Earth, right? So the chromosomes align at the center called the equatorial line. Next is, as seen in this image. Sorry for that. So we see that the chromosomes are already at the center, and we see that the centrioles are already at opposite ends with their spindle fibers attached to the kinetochore of each sister chromatid. In anaphase, we have a series of events that happen here. First, the chromatids separate from each other, and that place of union called centromere is dissolved. All right? So, the centromere is dissolved and the sister chromatids separate. Next is that we start to see each single chromatid being pulled by your spindle fibers towards opposite ends or towards the centrioles. All right? So each of the 2 sets of 46 chromosomes will be moving towards the opposite ends. So this end will get 46 chromosomes, and this end will also get 46 chromosomes. And at the end of anaphase, each set of chromosome will be able to reach the opposite pole, and the cytoplasm will start to divide. During telophase, the chromosomes will now be more visible to appear in 2 individual cells instead of 1 earlier. And as we can see, the nucleolus starts to form, and the nuclear wall or the nuclear membrane starts to appear as well. But what is most prominent here is the formation of the cleavage burrow or cleavage furrow, which is the separation of the 2 cells from one another. So, in summary, in prophase, the 3 events are the condensation of DNA to form your chromosome. Second event here is the movement of the centrioles to opposite poles. And the third event here is the dissolution of your nuclear membrane. In metaphase, the only event here is the aligning of the chromosomes at the equatorial line. In anaphase, 3 events also happen here. Number 1, dissolution of the centromere. Number 2, pulling of the spindle fibers to the opposite ends. And then third is when they reach already the opposite pole. And then in telophase, a few events also happen here. Number 1, the cytoplasm starts to divide, forming your cleavage furrow. Number 2, the nucleolus and the nuclear membrane starts to reappear. And number 3, they now have 2 separate daughter cells. So, in summary, or putting everything together, in interphase, this is how your DNA would look like. But in prophase, they become condensed, and the nuclear membrane starts to dissolve with the movement of the centrioles to opposite ends. Next is the alignment of the chromosomes at the middle. As you can see here, it's very visible. There is a darkening at the middle, and these are actually your chromosomes. And these are your opposite centrioles. In anaphase, the centromere has dissolved, allowing the sister chromatids to separate from one another, and the cytoplasm starts to dissolve. It starts to separate from one another, as we can see here. Finally, in telophase, there is now complete separation of the chromosomes, and a cleavage furrow is formed. And when mitosis is complete, we have 2 separate daughter cells already. Now, the last activity of the cell is differentiation. And when we say differentiation, it is maturation. No? When there is a problem in mitosis, it will lead to an abnormal proliferation of cells or an abnormal continual cell division, leading to the production of a tumor. When we say tumor, it means mass or bukol. Yan. So, tumors are formed due to problems occurring in the cell cycle. Some tumors are benign, while some are malignant. And when they are malignant, we call them a cancer. Malignant tumors are very dangerous to have because they spread by a process called metastasis. So in the exam, if you will be asked, what do you call the spread of cancer cells? You call it metastasis. So what happens in the formation of cancer? Let me just go back to a few slides. I just want to show you. Here we go. The problem with cancers is that they do not have a G0. Now, what is G0? G0 is what we call resting phase. So, after mitosis, it should enter G0 or resting phase. But in cancer cells, after mitosis, it will undergo again G1, S phase, G2, and then undergo mitosis again. So, it wants to undergo continuous or successive cell division. But the normal should be it should enter G0 or have a resting period. Okay? So let me just go back to the presentation. Yan. So again, going back to differentiation, when a sperm and an egg cell unite, they form a single cell called zygote. And then the single cell called zygote will undergo a number of mitotic divisions in order to produce the trillions of cells we have in our body. Now, these cells will not only remain as general cells, but they will become special cells. No? Some cells will become bone cells, some cells will become muscle cells, some cells will become brain cells, They will all have their own function. And that maturity and the formation of a specialized function is called differentiation. Okay? So during differentiation, some portions of the DNA become active and others become inactive so that a specific function will be activated. So some cells will again become your bone cells, nervous cells or brain cells, muscle cells, and red blood cells. So that's what happens in differentiation. Lastly, if we have abnormal cells, such as your tumor cells, our body will schedule them for apoptosis. What is apoptosis? Programmed cell death. So it is a normal process in which cell numbers are adjusted and controlled. So in order to prevent excessive growth, in order to prevent excessive size of certain organs, then they have to undergo apoptosis. Similar to a fetus, di ba? When a fetus, usually their hands are all connected, no? May web-like structure tayo dyan in between our fingers and toes. But when we are born, we don't have that anymore. So what happened to those connections? They are removed by apoptosis. Right? So even a fetus, when you look at a fetus, para siyang may tail. It appears like it has a tail. So what happens to that tail? It is removed by apoptosis. And in adults, in adult life, we continue to have apoptosis in our bodies. So we eliminate excess cells in our body. Also, just like our bodies, cells undergo aging. No? And there are various causes for cell aging, including your existence of a cellular clock. So just like a human, our cells pala, meron lang din siyang parang lifespan. It has a cellular clock. Second, when genes die, a certain cell also dies. And when the DNA is damaged, the cell will also die. Okay? So that's the end of this video. Hopefully you learned something. If there's anything that is unclear, kindly rewatch the video. Thank you guys.